Okay, hi everyone. My name is Evelyn. I'm currently a second year, just finished my second year, incoming third year PhD students in Memorial Kettering in New York City. So my research is currently is involving studying um, epigenetic regulation of anti-tumor immune response. I know it's a lot. I know there's a lot of strange words and really not many people actually understand that. So I'm writing down here. So my overall broad interest is in cancer immunology, which is a very broad field to say that we study how immune cells interact with cancer cells. By interaction, we means that, how do they see each other? How do they respond? How do they perceive? Do they ignore? Do they get the call? And with the overarching goal of trying to educate the immune system or to use our immune system to fight cancer. That's a simplest way of phrasing that. In addition, my previous research prior to joining MSK was working with antibodies, which is why I'm excited to talk to you about antibodies today. My previous lab, I worked in sort of like this engineering antibodies, how to make them better, to make them different, make them different cool stuff. Put drugs onto antibodies and use it as the imaging agent. So that's what my previous research was about. I think with the current setting of COVID, I think you probably have heard or seen a lot of immunology words and like phrases, and then you might wonder what that means. And I hope that after this class we can understand the newspaper a little bit better, more like scientific and not just just newspapers. So my interest other than research is mostly traveling. I also do a little bit of yoga. I cook. I think that's a lot of these activities are born out of the COVID, but traveling is still there, and dying to get back on the plane sometimes. But these are my email address. If you have any questions after this class today, you want to get in touch, you want to connect with me, these are my email address. Cool. Should we get started? Okay, so I think before I wanted to get an idea of what you guys know about immunity or protection, if you can type on the chat, maybe you could let me know what you guys think immunity or protection means, because that's really relevant to what we actually talking about these days, right? So maybe it's just kind of like
Okay, guys, so this is your do now. Type in the chat, what is immunity? What do you think is the meaning of the word immunity, or maybe the word protection? I want to see everybody typing something in the chat.
Can I say that? Do I have to type?
Yes. Oh yeah, you can say something. Yeah.
Okay. I mean, the word protection is another meaning of immunity. Is like so basically being protected from something, or being yeah, being protected.
So what do you think we're getting protected from?
Various things. I mean, viruses is one of them.
Getting sick, yeah, virus is really good, right? Things that make us sick, right?
Yeah.
So that's what we know most about the immune system, is that it's such a vague, broad, shield that protects our body from invasion from foreign things that are around us, right? So that's a really good definition to get started. And then I want to show you, how do I get to the next slide? So the immune system, that's what we think is a shield that fights. That's most people think about the immune system as fighting agent. But in reality, the immune system is such a complex network of everything that is in the body, different organs, different fluids, different things that have two main goals. One is to fight, prevent, and heal. I think many people don't think of immune system as healing, but it's actually very crucial to actually have that kind of balance in terms of fighting and healing that keep ourselves healthy and normal. So do you know what happens when the immune system is not really doing a good job of healing us, or it's too sensitive, or it's not, it's actually trying to attack our cells? You know what we get when that happens? You can unmute and chat as well, and you can type in the chat.
So we're looking at the question at the bottom of the slide now. What happens when the immune system is too sensitive? Anybody have any ideas?
It's okay if you don't know. That's totally fine, because it's not really the question we talk about usually. So what happens? I don't know. So we get allergies. We get allergies with autoimmune disease. We get things like lupus. We have things that, we sneeze out when spring has come. Some people can't eat peanuts. Some people can't tolerate milk or dairy. So that is when the immune system is too sensitive and it's not really doing a good job of healing. So overall, wanting to know about the immune system is not only to fight, as we most know in terms of COVID situation, but also prevent from a future attack, as well as heal us after we fight off an invasion. So the main topic today, when I wanted to introduce you guys, is antibodies, right? Antibodies is a part of the immune system, and I try to kind of look up on Google what it means when you guys look up antibody. And these are, for example, as Oxford languages say, that it's a blood protein produced in response to counteracting a specific antigen. So first of all, what is an antigen? Antibodies combine chemically with substances which the body recognizes an alien, etc., etc. Then I think this is Merriam-Webster. It says something more words, complicated words, and I feel like definition should make things easier to understand, not more complicated to understand. I think it defeats the purpose of trying to find something. So they're not wrong, but they're just very complicated, and it doesn't really orient us to a way to understand it. So I decided to kind of, this is where we are now. So we know that antibodies are part of the immune system. They're found in the body. It gives us immunity, that's protection, as we just talked about. And they work during infection or invasion by bacteria, viruses, what we just talked about, right? So these are where we are. So I hope by the end of the class, we can broaden each of this kind of question to a way that is more related to, scientific terms. So that's where we are, and antibodies are part of the immune system. But in fact, it is a protein that is secreted by a type of immune cells called B cells here, or plasma cells. Plasma cells is mature B cells. These are the cells that make up antibodies, because you just think of antibody as just like kind of like this little round circles that are secreted inside the body, and it's made by something, and that something is the cell. It's called B cell. And this B cell designed the antibodies to recognize a very specific targets called antigens. So antigens are the targets. I'm just going to refer them as targets for the rest of the class today, just for simplicity. And so the main role of antibodies is recognition. That's what we all need to know about: recognition, recognize. And it works as a key and lock system. As you can see, that you have the yellow key for the yellow lock, the brown key for the brown lock, and antibodies are the same. They are made to design, they're made to recognize and identify a very specific target, right? So, and then different antibodies, these are different classes of antibodies. They look different, but even so, you hear these like different IgG, ED, MA, they look different. So can you tell me what is the similarity? So what's a common thing of these different antibodies, of different classes of antibodies? Do you think you find something that these have in common? There's a hint. It's about the shape. It's about the shape. It's okay. So this is the next slide. You guys don't have to answer now. So the common thing with all these class antibodies is they are Y shape. They look like a Y. So basically that's what antibodies look like. And the first 6 things in the what you're looking at are just different ways that scientists usually draw out to identify antibodies. So the first one here is the most simplistic way, and most people identify, or when they draw antibody, as a Y. And it gets a little more complicated about the structure, what comes from, what it does, the chemical bonds, and all this cool stuff is getting more complicated. But what I want you to know that is part of the Y shape is very important because there's an arm, right? There's two arms and a tail. The arms of the antibodies here, as you can see, is what binds to a target or an antigen. So you see it engages with antigen here, and that's the arms. Each of the arm can also bind to the same or different antigens. And there's a tail. The tail part is also very important to interact with other immune cells, the components of the immune system. So it sometimes can work alone, sometimes work with others, but if we know, immune system is a complex network of a lot of things working together. So that's about the shapes of the antibody. So my next question is, can we pull up the poll question? Which part of an antibody is required to recognize a target, or which part of antibody recognize a target? Could you guys answer in the poll?
Take a guess, you guys. Take a guess.
So the polls are anonymous, so I won't know if you guessed it incorrectly or correctly. So if you see the poll on your screen, you can just choose one of the 4 options. And when about 80% of you all have done, I'll stop the poll. So I'll give you all a few more seconds. We have about 50% who voted. We're waiting for another 50%. So the options are 2 arms of a Y, the tail of a Y, 2 arms of an X, and I don't know.
That's totally a valid answer. A lot of times we don't know. That's what we are.
I wait for a few more folks to vote. I give you guys 3 more seconds. 3, 2, 1. Awesome. So we have a tie. So about 40% thinks it's 2 arms of a Y, and 40% thinks it's tail of a Y, and the rest of you all have no clue.
This is totally okay. Yeah, I know it's first time you're seeing this, strange Y-shaped things. Oh, how do I move this forward? Oh, here we go.
I can.
So I think about 40% you get it right, but it's totally not wrong that the tail is very important in terms of how antibodies work as well. But to recognize a target as its main function, right, is mostly at the arm. So the arms. So you think about it, it's like a bird person setting up with like 2 arms, holding up like that, and then the hands are what grabbing the target, grabbing the antigen. So the hands or the arms here is what recognize the target, and the tail basically in this situation is helping you stand, but in the antibody setting is also help with interaction. Okay, and let's so this is a very broad, super overarching question. I don't think you guys get it. So this is a pure guessing question. I think you guys have read the word neutralization or neutralizing antibodies when you read about COVID vaccines, or different kind of COVID vaccines are currently being applied. So I wanted you to know what the word neutralization means. But so try your guess.
Awesome. So we have 4 options on the poll. So the first one is neutralization is ingestion by cell eaters, and we call those in science macrophages, the things that just go around eating cells. Or neutralization means to kill, where you just burst the cell. And neutralization could mean protection by blocking access of this foreign thing into the cell, or I don't know. So I'll give you all a few seconds to answer. We have 1 person who responded, so we're waiting on the rest of the group. So does neutralization means eating the cell, killing the cell, or blocking stuff from getting into the cell? Here we have about 50% who voted. I'll give you all about 10 more seconds. We almost have 75% voted.
Great. I want to ask somebody who answered protection by blocking access to explain to me what that means. So and why did you pick the answer? So somebody can somebody tell me why they picked protection by blocking access?
So feel free to write your response in the chat and can give you about 5-10 seconds. If you're brave enough, feel free to raise your hand or unmute yourself.
For me, I break down the word neutralization. So the word, I feel like it is something related to where like somebody, it was, I just imagine where somebody is getting a injection where like they put alcohol on their hand to make it neutralized.
Oh. Wow,
So
that's very good. That's you're absolutely right. That's great. That's great. The word neutralization actually means what it means, right? It's yeah, I'm glad. But honestly, before I learned immunology, you to tell me neutralization, I would not know what that means, and I would not know that it's related to antibody or it's related to some class of immune components. So that's how do I? I'm sorry, I'm on this laser pointer that is making very, very hard to do this. Okay. So you're absolutely right. So I just wanted to go back to that when we talk about how, so antibody gives you immunity, right? So the way it does is either can prevent infection or help fight against inactive infection. So the parts that it help fight or it interacts with other immune cells is here. So when it engage with other immune cells, that macrophages that 36% that you picked, as it's actually ingestion, which means engulfment, like eating by macrophages, or NK cell, which is another type of immune cell that killed by lysis. So it injects something into the cell and the cells burst out and then die. So here is you have the image here is that you have an immune cell that engage with an antibody. The antibodies then engage with an antigen or a target here bound on an infected cell. So think about this, maybe it's viral protein, virus, bacteria, and the antibody recognize it. Then you have an immune cell coming and that recognize an antibody. So that's a very cool thing, right? So the immune cells or immune system interact with each other and help each other fight off an invasion. And then the cells that has the virus is become infected, is then died. A lot of you knew about this macrophages or ingestion, so that's very cool, right? So here is another situation when you have the antibodies here. As I said, in scientific, we always draw antibodies as a Y. And it recognizes little circles here that is on the surface of the pathogen, like the bacteria, a microbe here, anything that's small pathogen. And then you, what it does is it actually binds to that tail of the Y that we talked about. The tail of the Y here is then recognized by the cell eaters macrophages. And then what it does is it kind of like reach out its entire arm and engulf everything in it, and it digests. So that's one very cool way. And a lot of you already knew about neutralization, which is a process when antibodies surround a target, like coronavirus, right? And block it from access to a cell before it could cause an infection. That process is called neutralization. So it's basically, it's a basis of a lot of, you guys knew about COVID vaccines, right? That's what we have when we use our immune system to prevent an infection by a virus, and that what it is here. So you see that it blocks all access from this circle round shape here, which is an antigen or is a virus, or it can be toxin, it can be a lot of things, and it blocks it from access to a cell, so causing an infection. That is the process of neutralization. And the last part here, I'm going to go just to go really briefly because I don't think, it's not that important, but I think it's be cool to know that we think antibodies found in the body, and we think the antibodies work during an infection, but the reality is that we have different classes of antibodies, as I shown before, right? They all white shape, they look different, and they are very different, but they found in a different part of the body. So this particular strange X shape here that is made of two white shaped kind of pointing the tails together They're usually found in the mucus. So, basically in your respiratory tract when you sneeze out, and that kind of mucus when you're coughing, you sneezing, that is when these antibodies are found. Also in the lining of your stomach or your intestines, that's where these particular antibodies are found. However, most of us know antibodies probably in the blood. That's what, if you ask somebody, they will probably say antibodies in the blood. And I think this IgG antibody here is where it's most commonly found, and it's also longest lasting in the blood. So these different classes of antibodies are different, they're found in different parts of the body. And the very cool thing about IgG, for example, it's the most abundant because it's also longer lasting, and is the only antibody that can cross the placenta to enter the fetus to protect the baby during pregnancy. And in addition to that, you can see the IgG here is primary antibody that works during allergies. So you can see that antibodies can work in different settings, will be before an infection, during an infection, after an infection when you're clearing things off or you meeting a new challenge, or during allergies. So it's not really limited to pathogen, or bacteria, or virus. But we think of immune system as also part of any response to the foreign particles, or even without cells. So that's just the major ideas of antibodies. So in moving on to how antibodies are in the labs is what was a very cool thing to look at this. I found this figure online and I felt it was very comprehensive. A lot of us, when we work with antibodies, we work with different types of antibodies, and we make antibodies useful to us in the scientific setting. We make them the tools to use in the scientific research rather than just something that we in the body. So if you ask, do I take my antibodies from my own body and then study it? No, of course not. But what we do is we have commercial antibodies that are made elsewhere. They can be made in the lab. So the lab can also generate antibodies that are specific, again, for a specific target, for a specific purpose. So antibodies can be used in the lab as a tool for study to ask very specific questions. The thing that I'm going to focus on today, the part of the scientific experiment I'm going to show you today is mostly related to this major class of antibodies called antibody drug conjugates. So what that means is that you can see, can anybody, I think, let me just move this poll question, sorry. So what it means that in this antibodies you're looking at, it's in this red square here, that you see that in the tail of Y, it has some stars, some red or yellow stars. And that means the drug, or that means protein, that means something else that we scientists put onto the antibodies to make them useful for us. So they could be, for example, drugs. So we use antibodies to carry those drugs and then enter the cells, enter the body to go to a very specific place. Or they can be fluorophores for fluorescent proteins. What that means is just glowing proteins that glow. So it marks a certain colors. So that is useful for us too when we want to visualize something in the dark, right? So we want to see where things are from the microscope. We know where things are, so then we mark them by colors. Or as very cool things here, very cool part of it is that can also be linked to enzymes. So does anybody know what enzymes do?
Or if you know an example of an enzyme that you've heard or what you think it does, feel free to type it in the chat or unmute yourself. And if not, feel free to say you're not sure.
It's always okay to not know. It's always okay. That's very good. Wow. It's a really good, yes. Enzymes are just parts of, they create reactions. You're completely right. Wow, somebody, so speed of reaction, you're really good, because those are catalysts. So the enzymes are exactly what carry on a certain reaction, right? So what I'm going to introduce today with a mini lab is this particular technique called ELISA. I think ELISA is something that is very easy for you to remember, right? So ELISA, everybody can say that. But what it means is enzyme-linked antibody, basically. Immunosorbent assay, it's just a fancy way of saying we recognize something cool. Basically, what we use is that we have these antibodies that are linked to an enzyme. Particularly, you see in the green ones here, which we'll actually going to do later on together. But the idea of ELISA is that you want to detect a particular target. For example, you want to know if you have a pathogen, you want to know if you have a virus, you want to know if you have what your blood type is what you want to recognize, you want to figure out. So the idea of this is that you have an antibody recognizing it, and the antibody is actually coated on the bottom of a well, as you can see here and in the picture. And coated, it just means that you glazing something like a donut. So you have a sugar glaze on the donut, that's just coating. And then basically that sugar glaze is the antibodies coating the well. Then you interact with the antigen, or let's say here, the pathogen or coronavirus or anything that you wanted to see here is captured by the antibodies at the bottom of the well. Then you antibodies that recognize it, right? And here you can see that it looks a lot like neutralization that we saw, right? So the antigen is like sandwiched or captured between two antibodies recognize the same. Then there's another antibody that recognizes these antibodies. Do you see that the green white shape here is recognize the tail of this blue antibody here? And that green antibody is conjugated with an enzyme. The enzyme of this antibody catalyzes the reaction that changes the color of the well. So that changes of color infers that you have captured the antigen in the well. So there is something cool, interesting in the well, right? And if you don't see the change color here, that means that these antibodies have not detected anything. That means you don't have anything, so you're negative. Let's say you are negative for antigen of target ABO, that means you may be an O-type blood, for example. That's a different setting. So anyways, it's very simplified. It just means that you have no target in the well. So what we are going to do together, let me actually share the screen. My laptop on my iPhone. Can you all see my screen? So now I think, how do we?
Evelyn, you can stop the presentation and then I can spotlight your screen. There we go. All right, so I'm gonna, can everyone see through the camera phone?
Mm-hmm.
Awesome.
So what we're going to do is we're going to put gloves on, because you need to protect yourself, right? And then I'm gonna put my camera here. Hopefully you'll see my bench. You see here? Okay. So these are some ELISA wells that I have prepared yesterday. And these are what we saw from my slides. So you have the wells, and the wells here are coated with antibodies at the bottom. And I have put the antigen here at the target yesterday and incubated them. So hopefully it works. I've never done this overnight or the next day. So let's see if it still works. So this is the ELISA wells. What I'm going to do is that we have here color agent A, right? And this is color agent B. So we're going to mix these two together. These are the substrates of the enzyme. right? So we talked about enzymes catalyze or they create reactions, right? So the enzymes already on the antibodies here on the well, and we are going to make the substrate, and we're gonna hopefully see a reaction. So what I want to do is I'm going to use these black tubes here, and the reason why we have black tubes is because they when we mix this together, they become sensitive to light. So what I'm going to do, as you can see, these are different pipettes. They have different numbers here, denotes different kind of volumes. So what we're going to do is we're going to use this big pipette, has the biggest tips right here. And this is pipette, or this skinny round thing, is very useful in the lab setting because you can change the volume, you can use different ammo. So these are the tips. The tips look like this. They have a little bit of a filter. And then what we're going to do is we're going to mix and beat together. So this is, oh no, this is not right. So read the label, always read the label before you start. So what we're going to do is and then we take so when you become a scientist, you kind of know how to open and closing with one hand. So I'm going to mix things here. You can't really see the liquid in here. This is when you have to kind of trust yourself, and then trust that it's in here. What I'm going to do is I'm going to briefly mix this. And then what we're going to do is we're going to add 200 microliter of each of this mixed solution of A and B into the well. Hopefully, let's see. So a cool way of how to detect color changes is when you put it on a white background like this. And you see at the first row change color.
What color are you all starting to see in the well that looks like it's changing?
Light blue.
Yeah. What about the others? Do you see anything in the other wells?
No, that one I pretty weird.
Yeah.
So what we can do is we can cover this and go back to the presentation for a little bit of discussion, and we'll go back to here. This is a very hazardous waste. I haven't found a rack that fits this tube, so we're just gonna put them here and cover them, and then this tube should cross. So it's here, but we can go back into the presentation now and.
And while she goes back to the presentation, y'all can think about whether or not it will get a darker blue, if it's going to change to another color, or if it's going to go back to clear. Just keep that in mind.
Okay. Ooh. Can you all hear me?
Yes.
Okay. Cool. So I'm gonna put my camera here. In the next slide is a question that I wanted to ask you from the previous graphic that I showed you. What do you think? What principle antibodies allows for ELISA to work? So what is it serve as the basis of ELISA? So 1 is antibodies are specific to the targets. 2, an antibody can bind to or recognize other antibodies. And the last is antibodies can be linked to other stuff like drugs, fluorescent proteins, enzyme, or all of the above.
All right, so I just launched a poll with the same question. So what is the principle of antibodies in the ELISA? So the ELISA is the procedure that she just completed. And so the options are antibodies are specific to their targets, or an antibody can bind or recognize other antibodies, or antibodies can be linked to other stuff like the fluorescence, like the colors, different drugs, different proteins, or all of the above. So I'll give you a few moments to fill out the poll. So thinking about what she just talked about and what she showed, choose what you think best fits.
I want to open this paper here. Maybe it's easier for people to visualize.
Yeah, about 50% who voted. Wait for a few more, and then we can end the poll and see what the top answer is. Anyone else wants to take a guess? We have 60%. I'm gonna end the poll in 5, 4, 3, 2, 1. Awesome. So out of 70% who voted, about 60% of you decided that the answer was D, all of the above. Share those results.
That's a correct answer. All of the above. Very good, guys. So we know that antibodies are specific to the targets, right? So basically what it means is that these antibodies, the orange and the blue, recognize a specific red antigen here, not the gray ones, right? So it's specific to targets. And it can bind to other antibody. You see the green ones actually is bound to the blue antibodies, and they can be linked, or the conjugate is a scientific word for to other stuff, and that is what we saw that is linked with an enzyme that catalyze the reaction of color changes that we just saw in the well. Yeah, so that's just a very simple basis of ELISA. Now let's one application of this principle ELISA, as we can see, is blood tests. So we know what antibodies are, what they do, how they do it. Then we started to understand how they applied. Normally not in only the lab, but in hospital and different research and medical settings. So what it's blood tests actually is they have different antibodies against different antigens, right, or targets. So here you have a red blood cells. You have different red blood cells here. They are bounded. They're bound by different antigens. So A antigen, B antigen, A and B, or none. Then what we do is we detect those antibodies using specific antigens against either B or A. So basically, what we saw here is that if you have blood 0, then you have no antigens, right? So when you have anti-A, anti-B, you don't see the bound. But however, when you have antigen A, for example, and you use antigen B, it will not recognize, and vice versa. So that's basically just an application of what it means when we understand antibodies, what is in the detection and diagnosis kind of setting. So this is the last question I have in the class. We can talk about this before while we're waiting for the well, but this is a very open-ended question. I think it's like the first question I saw with the class. it's kind of, to for you to a little bit further your thoughts about antibodies. So if your friend caught virus X, we're not going to say the name here, virus X is whatever virus, and your friend has also caught virus X, does it mean both you and your friend have the same antibodies? And then you can
All
type
right.
into that.
So I'll give you a moment to digest the question. So Evelyn's asking if you and your friend both caught the same virus, and would you both have the same antibodies? All right, so you can put yes, no with an explanation in the chat, or if you're feeling brave, you could unmute and say it.
Okay.
It's definitely not a straightforward question. So
I
take your time. Meanwhile, I'm going to check the web really quickly, and I can just bring it over here.
Go ahead, Ujwal. Ujwal, is that how you pronounce your name?
uh-huh.
Yeah.
So I feel like the answer is no, because since antibodies are like things that our body produces inside the body, right?
Mm-hmm.
Uh, and depending on, even this virus could be same, but the genetics or of the virus can be different. So based on that, our body can like the mutation, you could call it, and our body can have to produce similar kind of uh,
Mm. Okay, I like where you're going with that. And Laura
antibody
mentioned,
fight the virus. Okay.
sorry, Ujwal. And so Laura also mentioned that antibodies work different in different bodies, also because blood type is not the same. So maybe what they produce is different. All right, I like how y'all are thinking. Anyone thinks that it's going to be the same? Who wants to debate?
So what is while we are on this, I'm going just to cover it as well. I think we should, by the end of the class, we should be able to see something cool.
Awesome. So I'm going to assume most people think that the antibody may be different, even though you're infected with the same virus. So Evelyn, tell us, what do you think?
You guys are right. You guys are getting there. So what the question meant was to ask is whether, we talked about specificity, right? We talked about different interaction with other parts of immune cells. We talked about, antibodies can be made to do many different things as a scientist. But when we go back into the question of, our cells, what it means to translate this question to us, when you caught, let's say, virus X can be a part of the pandemic, right? And a lot of us got the same virus. Essentially, the antibodies will be different. They, first of all, these antibodies, even though they recognize the virus, they recognize different parts of the virus, right? So they can be different parts. Even if you have the same antibody as your friend, because you might have some of them are similar or the same, some of them can recognize the same part of the virus. So you and your friend have a lot of antibodies, but maybe one of them is similar or the same. What similarity here is when we talk about recognition, but the different part here is mostly in the tail or the rest of the antibodies, right? So two antibodies can recognize the same target. Does not mean they're the same antibodies. That's the idea of this question. So if you have antibodies recognize the same target, they can be completely different antibodies, and that's in the lab, we know these antibodies are different because maybe they're made differently. They are made of different genetic makeup. So one of you has said, touch on the genetic or the mutations, right? So antibodies are very, very diverse, and they are made to have a very diverse recognition of different parts of a virus, different parts of a protein or bacteria. And even if they recognize the same, the rest of them would be different. So the answer is no. So what I want to take you to take away with this question is that if you have antibodies recognize the same target or the same antigen, they do not mean that they're the same antibodies. That's just the overall wrapping up of this question. And now I want to go back into the well, and then we're going to see this reaction, how it's going, right? We are seeing this enzyme reaction, right? And then we're gonna see whether we see any change in the colors.
Evelyn, you can end the presentation so we can see your screen with the wells. Yeah, perfect.
Okay, can you all see my
Yeah, we can see the table.
so let me just turn off okay. So
Ooh.
can you all see it?
All right, tell us what you see. Oh, the camera went off again. And there we go. So the well to the far left, we're going to call that 1. The one in the middle, we're going to call that 2. And the one at the right, we're going to call that 3. Can anyone tell us what do you observe?
All of them had different color.
Mm-hmm. So they're all different colors. Does anyone
So
want
you have
to guess as to why they're different colors?
yeah, that's a very good list. I was just gonna get there. That's very good. So do you know why they're different?
Is there more of something, less of something? You're not sure. Yeah, the colors are more deep, you know?
So I can read your answer.
You can unmute yourselves or type it in the chat.
There's a possibility. I don't remember.
What we are going to do, because this reaction will continue going on. So what we're going
Ujjwal
to
was
do.
gonna say, sorry, Ujjwal, you were gonna comment on the colors?
I was just saying, because of like first we use 2 different type of enzymes, right, in order to make the lab. So because there could be a possibility because of it, the color is different.
Okay, so you think it has something to do with the enzymes?
So I think you are right. So what happened here is that I use the same enzyme basically for all 3 wells. So they're not different enzymes. The only part that's different in these wells is that the first well here has the most antigens. So it has a lot of antigens here. The second well has less, and the last one has no antigen. That's when you see that ELISA is a very sensitive technique that detects the amount of antigens in the well, or the amount of targets in the well. So this is what you see is the reaction that is carried by the enzymes bound to the antibodies that catalyzes the reaction chain. So in the lab, we also have something that is the stop solution. So what we're going to do is we're going to stop this reaction. We're adding
you using the wood? Oh, sorry. No. Is there anything in there? No. Okay. No.
It is magic. I think of science as magic all the time.
So the reason why we need to have the stop solution here is we want to stop the reaction, right? So we don't want it to be too blue or too, like too, so basically we don't want it to be too dark of a blue. So it's like darkens up. And it changed the color to yellow here. So we would enable this to be detected with a machine. So this actually can be read on a machine in a very specific wavelength to read out the certain number. And this number correlates with the amount of the targets in the well. So do you see that? So basically what it means here is that yellow means that yes, antigen, more yellow is a lot of antigen, and then white here means nothing in there.
Yeah. Awesome. I have a quick question for you guys, because I also think about it when I think of ELISAs. So if you had 3 people who were infected, one person got infected on day 1, and you checked them on day 1, you checked them on day 10, and then you checked them before they were infected. Which one would be darker, the day 1 or the day 10? Yeah, Laura said day 10. Mark said day 10.
My ELISA was.
Yeah, right. So you're thinking the longer you're infected, the more virus is replicating in your body, or the more antigens are present, so the antibodies combine. You guys are on it.
So sorry, I need to turn this on. I'm gonna leave this meeting here. If you guys still need this, I'm gonna show you guys here. It's here. Well, sometimes we don't use it. I'm sorry. Right. So that's just how ELISA works essentially. Because of the time that we have, I wasn't able to carry the entire 6 experiment or the technique. It would take like some time to like 5 hours. So we have kind of condensed it to kind of like half an hour, 10 minutes kind of cool color changes. And that's one very sensitive or one really cool thing about antibodies, that they are specific, they're very sensitive, and they're really stable. So that's what we use in the lab. And I think I was going to show you a tour of the lab.
Estaba too expensive. Un café por $15. Demasiado plata.
Pero un café.
¿Qué?
I think they accidentally unmuted.
Yeah, I was going to give you a tour of the lab, but I think we don't have time. But before we leave, I just wanted to chat a little bit, as you guys are in high school and I was in high school once, and that was when I got excited about this stuff. So when I was in high school, as you guys, I was, in a lab for a summer. And that's when I first had my first exposure to science. Back in the day, we didn't have this kind of Zoom, classes. We didn't have this different kind of internet enabled classes. So we kind of have to see a lot of things in person. And I worked in a lab back then. What we did was we made a low-cost H5N1 vaccine. I know H5N1 sounds so long ago. But it was once another part of the pandemic and the respiratory virus that make us sick, and it was very long ago. But what we did was we made vaccines low cost, so using technology, and I was amazed by everything that I saw. Everything that I saw, I still remember. I drew out thing on my notebook, and my notebook, I still have that, is pink, and it's like super, it has like a different pink hearts on them, and then it's me, like pictures of like scientific tubes and like things that words I don't know, because I'm afraid that I will forget about them. That's why I have to write down everything. And then, in the end, I was like, once you embark on this field of science, that these are things you need to know. But back in the time, it looks like magic to me. It looks like, why do we need these different kind of tools? And if you guys are interested, in science and want to pursue as his career in the future, what I would think is the first thing you need to have is curiosity. And I think that any scientist will tell you that, when they stay on this path long and longer, they remain curious. They're curious of different things, of different questions, and they pursue those questions. So that's why, asking questions and thinking of questions as a very important part of science. And science can be different things. It doesn't have to be biology, as we talked about today. It doesn't have to be immunology. It doesn't have to be cancer. It can be, chemistry, physics, any other parts that allow us to think critically and creatively. So if you have, the creative aspect and you're curious, I think science would be a great way for you to embark on in the future. With that.
Any questions for Evelyn? How did she get into science, the work that she does? Any other related/unrelated questions? Feel free to write it in the chat, or you can unmute yourself and feel free to ask. Any questions on antibodies or ELISA?
Sorry, my lab mates are walking around carrying boxes, and then things fall off and stuff, and it's loud and it's chaotic. That's what happens in the lab. It's an open space. So you see my coworkers, like my other grad students here. It's like, what are you doing? So it's a very open and collaborative space.
So Laura wants to know, what's the difficult thing to be a scientist?
I think it's a lot of studying. Now I think back about it. It's being a student, right? I think a scientist is a student of science, and even though we talk about it and we teach and we present and we publish and we do all those cool things, we discover. Essentially, we are students, and studying is part of becomes the difficult thing and into a passion. I think in the past, when I was younger, I didn't actually have the patience to actually study. I was, a spontaneous person. I was not into discipline. I don't like being told what to do. I don't like principles. And, I was not made for science at all. I studied arts for the longest of time. I almost applied to art school when I was in high school, not made for science. And then, so the difficult thing is kind of like get yourself to study. And I think what changed in me was, the experience I got myself exposed to. It could be what you read, it could be what you watch, it could be who you met and the people around you, and that inspire you to want to learn. And I think that is the most important thing, that it gets you on the path of studying. And for me, that's the most difficult thing. This is long hours of studying, but it's knowledge becomes something that you want to pursue, and because of that part of studying, you might have to sacrifice a lot of things. When I was in college, for example, my convocation day, which is like the first day of school, my senior year, instead of going with my class to convocation, senior college, right? We all like dress up and the entire school like honors us being senior and we're graduating. I was in a mouth surgery for 7 hours, and I did not go to convocation. So that was something that you learn to do, and then you want to do. So and that, I think that's something that's inherent about it, and the spontaneity and the grit of studying. That's the most difficult thing, but the reward is great. So the reward is incredible. So for example, I always tell my friends this when I talk about what I do, is that sometimes if you become a scientist and if you are discovering something new, and at the moment when you discover it, for example, you knew it or you saw it, think about you're maybe the first person in the world that knows what you know, right? And then that's something that is the kind of, we call it the very high, and the discovery part and the learning and the knowing and the truth that it implies, because we can talk about politics, we can talk about all these ideas, but science, it's for me, it's truth. It's truth that can be replicated. It's truth that is the basis and the foundation of a lot of things around us, of the human body, of the the house, of everything that is true. So that's a reward. But yeah, the difficult thing is a lot of studying if you're not a good student. I mean, I was not a studious student in my class either, so.
I had one more question. What college did you went to?
In high school and college. High school, I assume, right? Because we are all.
No. What college did you went?
Oh, college that I went to. I went to Mount Holyoke College. I know you guys knew the college is in Western Mass. It's where the same neighborhood with Amherst College, Smith College, UMass Amherst. It's in like a mountain in the western part of Massachusetts. That's where I went. Yeah, but I did most of my research. I did at UMass Amherst actually. So I think UMass Amherst might be a bigger name that you might recognize. Mount Holyoke is kind of small, but that's where I went to. And I honestly, I tell you, it's I wasn't actually into biochemistry at the beginning. I actually wanted to do something differently. So I was a pre-med student, and then I decided to pursue dietetics and nutrition for a couple of years. And then I ended up also was studying English literature at the same time. So I ended up, being this weird combination of biochemistry, nutrition, and English minor student with a thesis on something related to macrophages and breast cancer. Something like, that's what happens in college. You find something you like, and then you wanted to pursue it, and then there's a couple of classes, and bam, you're a minor in English, and that's that's what it ends up. But yeah, I was a biochemistry major in Mount Holyoke College.
Nice. Any other questions?
I would love to talk about anything if you guys wanted to reach out to me. Yeah, I also talk every summer. I teach a similar course for high school students in Vietnam. So I teach. I'm sort of. I was born in Vietnam. I grew up here in the U.S. So I'm sort of bilingual in Vietnamese and English, more English than Vietnamese. And what I learned about science and biology, I learned in English. So what we ended up is we teach the Vietnamese high school students in Vietnam as well. So teaching is also my passion, and mentoring is my passion as well. So if you have any other questions related to your career, I cannot answer your math question or your biology homework or chemistry homework that I don't think I could do. But anything related to what is going on in the field, or like what does this mean? Then yes, I'd love to talk to you guys about. So with that, I think I have, so these are my email address again. If you wanted to reach out to me, this is my email address.
Awesome. If there aren't any more questions, we can wrap it up. Thank you all so much for coming and participating. Hopefully you learn a thing or two about antibodies going forward. So now when you hear it on the news or you see it on Facebook, if you all do Facebook or Instagram, I don't know what are you, or TikTok.
TikTok.
Now you have an idea. We have another question. Lizette, does being a scientist take you a lot of time? For example, sometimes the doctor don't have much time for them.
I think so. I think the different about one of the different thing about being a scientist and a doctor is yes, it takes a lot of time training and practicing. So I think those are both careers that require learning and practice. But in terms of training that can take you, for example, PhD 4, 6 or more years. So some people do 8 years, some people do 4 years, some people do 3 years. That's after you do your college, 4 years of college. And in terms of a day-to-day, a scientist can work between 3 hours or 20 hours a day. Depends on the amount of work that they need to do. So I think both are time-consuming careers, but the difference is that I think personally, I think scientists have more liberties of deciding on the day, deciding on the time, defining their schedule. It's especially work in a setting like this, and in a setting like in universities or in nonprofit hospital like this, and you are more in charge of your day. You can come to work, and then, for example, I have my coffee in the morning that I made at home. I come in, I come in at 9:30, I come in at 9, come in 8:30, 10. But then the day takes me to where I need it to end. I think doctors probably work more hours compared to scientists. Yeah, and then some doctors also scientists, so they also work in research as well. So they have both the patient, the clinical side, and then they also work in research. They're also scientists. So yeah, it really depends on the type of work.
And one fun fact about being a scientist, if you want to go into school after college and you want to go to grad school to get a PhD degree, which takes about 5 to 7 years, if you do it in biomedical research or biology or one of the sciences, you actually don't pay for school. You get paid to go to school. If you go to medical school, it's different. You have to get paid. I think the reason why I love being a scientist is that you also have the flexibility of doing many things after school. So you could decide to still work in a laboratory, you can decide to work in policy, you can do consulting, you can work with industry, so like Johnson & Johnson or Pfizer or Moderna. So there are a lot of different options that you can do. And you come out of school debt-free, depending on what you did for undergrad. But it's definitely a perk with doing a PhD in science in comparison to doing an MD. So keep that in mind if you're thinking about going into STEM. There are many different options outside of being a medical doctor if you love science.
Yeah, I think both have good perks, and they are the careers that very involved in discovery and practice and learning new things. There's also path of the MD PhD. You can do both. You can be a doctor and a scientist at the same time, and that's also free. It takes between 6 and 8 years in addition to college. So the many paths is open to you. You don't have to get a PhD to become a scientist. People will get a master's degree. People don't have a master's degree and they decide to work in industry for a couple of years first and be a scientist there. There are many ways that you can be involved in science and be a scientist. But yeah, so it takes time definitely after you graduate, but yeah, it's more of like a journey than a career.
Yeah, awesome. Well, yeah, thank you all for your questions. Thank you for participating. Thank you, Evelyn, for such a great presentation. Thank you, Ruth, for signing up. And
Thank you for joining.
Yeah, thank you. All
Thank
right,
you all.
enjoy the
Have
rest
a
of
good day.
Friday.
Thank you.
Enjoy the weekend.
Enjoy the weekend. Bye, guys.